what's really cool, I think, about Andrew's work, even though he has an H-index of 33, which is very impressive for somebody of his age, is that he's probably had even more impact in terms of his bioinformatic and chemoinformatic tools that are accessed by individuals using the web. And those include BioGPS and Gene Wiki, just to name a couple, and I'm sure we're going to hear a lot more about those resources today that have really changed the way people do research. So Andrew, it's a privilege to have you as a colleague. I think this large and diverse audience reflects how many people Andrew collaborates with and enables their research. So welcome.
Thank you very much, Jeff, for that very generous introduction. So I am going to talk today about knowledge management, and my biggest challenge is going to make that dry-sounding subject relevant to all of you and try to convince you that it's critically important for how we deal with genomics and precision medicine moving forward. So I think especially in front of the home crowd, one would love, or I would love to tell all of you about all of the activities that's going on in my group, but of course that's just not possible given the time constraints. So I hope you'll indulge me for one slide just to tell you what I'm not going to cover in this talk today. And I think it is important because these overview slides, I think, establish some of the synergies and that may be apparent or emerge out of collaborations with folks in the audience. Okay, so conceptually and largely arbitrarily, I draw the activities in our lab into two sides. On the left side, we have biomedical discovery. This is where we really focus on that discovery process. This is largely done in collaboration with domain experts in other areas. And this is our rough, partial list of some of the collaborations that we've had. And I'll just say that, in summary, that this is one of the great reasons why I love being here at Scripps, the collaborative atmosphere and the opportunities that produces. In the area of bioinformatics tools, I'm going to further subdivide this into two areas. So we have the area of actually building infrastructure on which other scientists, both bioinformaticians and genomic scientists, can build. And we have a variety of tools listed here, and they range in terms of usage from getting millions of hits per year to millions of hits per month. So I think they're quite well used resources. I'm actually going to not talk about this, in large part because Chun-Li Wu, who's somewhere in the crowd over here, an associate professor of MEM, we've worked together over 10 years, and Chun-Li has really established himself as the intellectual leader in this area, taking things further than I could have taken it, and now even in terms of independent funding streams. So if you're interested in more in this area, take the next opportunity to chat more with Chun-Li. The other axis of bioinformatics tool really focuses around our activities using crowdsourcing. Crowdsourcing simply is developing platforms where we can collaboratively tackle big challenges, and we have a variety of different projects in this area. Similar to the role that Chun-Li plays down here, Ben Good, an assistant professor of MEM, plays a leadership role in many of these projects. And I should say that both of these individuals have independent projects that even aren't shown on this slide. And then, of course, there's filling in the gaps, the synergies between these three various areas. And I want to especially say that, the bridges between biomedical discovery and our bioinformatics tools are especially important. These are what keeps us anchored in things that really matter. And so we aren't building tools for tools' sake, but really build tools that advance biomedical research. Okay, so that's the overview. What I'm going to talk about today are two projects. In the crowdsourcing biology, I'm going to talk about our Gene Wiki and our Wikidata efforts. And then I'm also going to touch on our Mark the Cure project and its relevance to NGLY1 deficiency and rare disease in general. Okay, so let's start with actually rare disease. So rare disease is defined as, in a decidedly US-centric definition, any disease that affects fewer than 200,000 patients in the U.S., so less than roughly 0.1% of the population. And although individually these rare diseases may be rare, not affect many people, collectively they affect many people. So 30 million people, or roughly 1 in 10 Americans, are affected by rare diseases. And so to really illustrate some of the challenges, I think, that exist in rare disease research, I thought I'd start with 2 case studies. Okay, so case study number 1. Both of these case studies, the individuals involved have been very open about their diagnoses and their families' experiences. So this is the Beery family, Alexis and Noah Beery. These are 2 individuals who are fraternal twins who, when they were young, were diagnosed or had many phenotypes associated with seizures and developmental delays, had various missed developmental milestones. They were diagnosed with a disorder called dopamine responsive dystonia. Okay, and they were treated with L-Dopa as kids and responded quite well. However, in their teens, they developed actually some life-threatening side effects and symptoms. And so this was right around the time whole genome sequencing was emerged. And so they underwent whole genome sequencing. So based off the presentation of their symptoms, it was assumed that they had a mutation in this tyrosine hydroxylase gene. And this tyrosine hydroxylase gene is upstream of the production of dopamine, which explains why L-Dopa helps symptomatically. But after whole genome sequencing, it was actually found that they had a compound heterozygous mutation of this upstream SPR gene, sepiapterin reductase. And this was interesting because sepiapterin reductase is upstream of both the production of dopamine as well as the production of serotonin. And so based off of this biochemical map, the clinicians and the genomic scientists involved suggested that they could benefit from treatment by 5-hydroxytryptophan, which is an over-the-counter supplement. And in fact, after a couple weeks on 5-HTP, the twins returned to being essentially asymptomatic. So I would classify this as a pretty stunning story in genomic medicine and rare disease research. This is a picture from a year and a half ago, I think, of the Beery twins. They were off to college in the fall of 2015. One of them is, in fact, a college athlete. So, rare disease case study number 2. This is the Mite family. This is Matt Mite and his wife Christina and their 3 kids. This is Bertrand Mite, who also presented with a variety of phenotypes as a young kid. Developmental delays, seizures. Some of the most notable symptoms was his inability to produce tears. And they, for a very long time, were on a diagnostic odyssey trying to learn what disorder that their son had. This is outlined in a fantastic New Yorker article. If you haven't seen this, it's called One of a Kind. Really an inspiring read. They, too, underwent whole genome sequencing. And what they found was actually that Bertrand had mutation in this in the gene encoding N-glycanase, the N-gly1 gene. So N-glycanase is responsible for cleaving off N-linked glycans of unfolded proteins so that they can be degraded by the proteasome. The presumption is that loss of function of N-glycanase function leads to aggregation of aggregates in the cytoplasm. So this was what they learned from their whole genome sequencing, but unlike the first case, it led to actually no obvious treatments. And the question is, what separates these 2 case studies? What made the SPR case so successful and N-gly1 so difficult? There's a variety of reasons why this could be, and I'm going to suggest that one reason could be that SPR is on this nice landscape of a biochemical pathway, right? So we understand its neighbors and its interacting partners, and it's akin to something like the Google Maps of the landscape around N-gly1, around SPR, whereas N-gly1 is much more of a cloudy road, and we're trying to find our way through this area of biology that has been less well completely described. So if you put yourself in the shoes of Matt Might and Christina Might, and you say, okay, I've learned that my son has deficiency or mutation in NGLI1, what do I do now? Well, if you go to PubMed at the time, you could actually search for NGLI1, and you would find all of 11 articles written about NGLI1. And you could probably, read that in a day or a few afternoons. But to round out what's known about Bertrand's symptoms, you might also want to explore this very unique symptom, the inability to produce tears. And there you get 164 more articles that might possibly be relevant to NGLI1 deficiency. You would want to look at what's known about the NGLI1 version in other organisms, also known as PNGAs. What can we learn from model organisms that may influence our knowledge of mechanistic information? This is one in a class of congenital disorders of glycosylation, which may be relevant there as well. This, of course, is related to this ER-associated degradation process, which has a richer and richer literature around it. And you could keep going on. You could go for genetic interactors of NGLI1. You could look at glycosylation in general. You can look at all the other symptoms. And pretty soon, you start coming to the realization, of course, that biology is incredibly interlinked, right? And any one of these 25 million articles in PubMed could be potentially relevant for understanding NGLI1 deficiency. So on the one hand, I think we can take some amount of pride and some amount of hope in the fact that the biomedical literature is so massive. Okay, so this just shows the number of new articles published each year. We're currently at over 1 million new articles published each year. That averages out to one new article every 29 seconds, every single day, every single hour of the year. So although the literature is massive, I would say that, unfortunately, it is pretty hard to query and compute on the sum total of our biomedical knowledge, right? So if, for example, I was interested in this query, I wanted to look for tyrosine kinase inhibitors that are used in hematological cancers. And if you were to search for those two phrases in PubMed, you get all of 496 articles back. Okay. Now, it's clear that tyrosine kinase inhibitors have a long storied history in terms of their use in hematological cancer. So why is it that we only get 500 articles back, right? It's because PubMed effectively is just a keyword-based system. So to do this query correctly, you would actually have to enumerate all the tyrosine kinase inhibitors that you're interested in. You would have to enumerate all of the hematological cancers that you are interested in, and you would have to do that pairwise query. Okay, so clearly not an efficient use of essentially all of the biomedical literature and the biomedical knowledge that we've accumulated. So I hope to some extent I've convinced you that biomedical research relies on effective knowledge management, right? It isn't to say that, for rare disease or common diseases for that matter, that all the answers are still waiting there in the literature and all we have to do is organize it well. But I think it is not a far stretch to say that science is most effective when we build off of the sum total of accumulated knowledge that is known up to that point. And so I would argue that currently we don't take very good advantage of accumulated knowledge. Okay, so if we think about, again, the sum total biomedical knowledge, all of the biomedical literature as a resource, the question is how do we better organize what's known in that corpus? This relates to essentially in library sciences, a process called information extraction. And we can boil down information extraction into two steps. First step being identifying the important biomedical concepts in that text. So if I have an arbitrary abstract here, I might want to understand what diseases are mentioned in this particular abstract. I want to highlight what genes are mentioned. I want to highlight what variants are mentioned, maybe what drugs are mentioned. Okay, so there's a handful of types of entities that would be important to categorize for every article. And then the second step would be not only identifying the concepts, but to identify the relationships between those concepts. So we want to know that this K509I is a mutation of KIT, and mutation of KIT causes this familial systemic mastocytosis. This is treated by imatinib, and so on and so forth, right? So we want to know the relationships between the concepts as well as just the concepts themselves. And of course, we don't want to do this for single articles. We want to do this for the literature en masse to create this giant network of interconnected biomedical concepts that has these properties here. That is comprehensive with respect to what is known in the literature, that it's current, so it's kept up to date with this 1 million new articles that are published each year, that it's computable, right? No person is going to be able to sort of browse through this network, so we need to make it computable so we can build visualization and analysis tools on top of it. And finally, it's traceable, right? I want to be able to identify the evidence behind any one of those edges that is in that massive network. Okay, so this is a grand challenge. Clearly, it's a challenge that many people have articulated, and really it requires the efforts and talents of many different disciplines. So you could imagine that it really takes a team to do this. Imagine the collaborative efforts of people in the biocuration community, people who do text mining and natural language processing. Ultimately, we'll need to get publishers and funders involved, and so on and so forth. So our particular approach to addressing this grand challenge is to use crowdsourcing. Okay? And again, crowdsourcing is this idea that we can build platforms that enable people to collaboratively do big things together. So one of our first entries into crowdsourcing, stepping back a little bit, was this project we coined the Gene Wiki, and roughly back in 2008. And the idea was simple, right? The most successful crowdsourcing application of all time, I think, is indisputably Wikipedia. Okay? And so the goal was to leverage the dynamics and actually the infrastructure behind Wikipedia to create a review article for every human gene. And importantly, this review article would be collaboratively written, it'd be continuously updated, and it would be community reviewed. Not peer reviewed in the traditional sense, but community reviewed, just like all of Wikipedia is. So, so the way we approach this is that, well, we said that we can identify bits of structured information from databases that we all know and love in the community, right? We can get protein structures, we can get symbols and identifiers, gene ontology annotations, so on and so forth. All of this we can mine en masse using sort of the bioinformatics integration approaches that we already have that many of us in the community use. And so the idea was, well, then we could just simply reformat that a little bit, slap it into Wikipedia, and create these pages for essentially 10,000 genes, or essentially any human gene, en masse. And the idea was that if you provide a landing spot, right, that people will be able to get to when they do their Google search after they've done microarray or an RNA-seq experiment, then you actually get to this page where they can find useful information. And that some proportion of the people who come to this page will say, yeah, this is great, but I also happen to know X or Y or Z. And because of the dynamics of Wikipedia, where anybody can edit Wikipedia, then we can continually build up the knowledge of these contained in these review articles over time. So this initiative, again, started in 2007, 2008. It currently gets collectively about 4 to 5 million page views per month over these Wikipedia articles. And just to show you, what we consider, I think, a success story, although much of the page on this, much of the content on this page for Relin actually pre-existed our effort. But anyway, there is to say that Relin has a long introduction. There's actually a table of contents. This Relin page goes on. And just to highlight one excerpt, the types of information you get would be, something like this, right? The expression of the protein has been found to be significantly lower in schizophrenia and psychotic bipolar disorder. Okay, so this is both the advantage and the challenge associated with Wikipedia. So any of us as humans can read this sentence and we can understand the the significance and the context of that statement. But that's actually really hard for a computer to read. So now suppose I wanted to ask the question, well, tell me all genes or proteins that are downregulated in schizophrenia or in bipolar disorder, right? Short of reading every Wikipedia article or, again, going back into the 25 million articles in PubMed, you can't computationally do that query, right? Natural language processing is good, but it's not that good. In theory, you actually just want this to be a database query. I should be able to ask, well, what are all the genes that are associated with any given disease? Again, there are such databases that exist, for example, that have these gene-disease associations, and I've just highlighted 4 of them here. And just to illustrate the point, though, if you go and now look for relin in any one of these genes, so if you start in KEG, right, you actually get this, a single entry, right, single gene disease association for lissencephaly. If you go to OMIM, you also get lissencephaly, but you also, in addition, you get this familial temporal lobe epilepsy, okay? So that's good, but we haven't seen our bipolar and schizophrenia. Thankfully, if you go to PharmGKB, they don't even categorize these 2. You get 2 other ones. You pull in schizophrenia, as well as autosclerosis, but still no bipolar. And then finally, if you go to this database run by the CDC, you actually get many of these, including schizophrenia and bipolar disorder, but in the context of 27 other quote-unquote diseases, which might include memory, short-term memory, weight gain. So, I mean, it's semantically all mixed up in terms of what's categorized here. So you can see that this idea of having databases that encapsulate some of the key knowledge we use in biomedical research, right, we're very, very far from having that organized in systematic ways. Okay, so I'm going to step into the most current area of research that we've been pursuing in this area, the extensions of the Gene Wiki project, into a system called Wikidata. So to explain Wikidata is actually best explained by analogy. So Wikipedia is to text as Wikidata is to data, right? So Wikipedia allows massive crowdsourcing to produce text that is human readable, and Wikidata enables massive crowdsourcing to produce structured database content so we can do the queries of the type that I mentioned before. The mission statement is articulated here really well, I think, by the founder of this initiative, and it's to provide a database of the world's knowledge that anyone can edit. We extended that, just like we sort of hijacked Wikipedia for these biomedical research terms. Our goal is to seed Wikidata with as much biomedical knowledge as we possibly could as a starting point. And then that will allow us to grow and nucleate downstream contributions from people all across the community. Okay, so now let's go over to the Wikidata page for relin. Okay, so now, although this is a web page here, it essentially represents the structured content that is available for relin. Since this is incredibly small text, I've just isolated it out. So everything in Wikidata can be boiled down to a claim, right? Or it's often called a triple. So relin is a subclass of, or is a type of, protein. Relin physically interacts with the LDL receptor and amyloid beta A4 and so on and so forth. So there's always 3 components: a subject, an object, and a predicate, a way that the subject and object are related. And down here, you can see that relin has decreased expression in schizophrenia and bipolar disorder. Okay, so importantly, right, these aren't just words, but every one of these concepts, every one of these boxes or terms is related to an unambiguous identifier. And this is important when we talk about data integration, because data integration, it relies on the fact that different resources are pointing at the exact same concept. Okay? Again, this is a web page that encapsulates this information. For the programmers in the room, it's important to know that this is all also available in a structured format, right? So there is an application programmer interface, an API, where you can query this, and you can get all this information in a structured form that is appropriate for computational analyses and tool building. And getting, again, back to the very specific thing of decreased aggression in schizophrenia, just to say that the Wikidata data model also has a couple other things built in that I think are really important for biomedical research. There's this concept of qualifiers. So here we're saying that this was determined by immunohistochemistry, and it could also be, a qualifier could be only found in a certain cell type or only found in a certain population. So it allows you to encode in a programmatic way some of the qualifiers, and then also the reference, right? So you can go directly to the evidence where you can, where this is asserted, and you, as a scientist, as a domain expert, can evaluate that and weight it as appropriate. Okay, so again, our goal is to seed Wikidata with biomedical data to nucleate the crowdsourcing effects that we saw with the Gene Wiki in Wikidata. So through the efforts of many individuals in our group and our collaborators, we've now loaded all human and mouse genes and proteins, all gene ontology terms, all FDA-approved drugs, all human diseases, or all human diseases in the disease ontology, and then as well as quite a bit of work in terms of microbial genomics. The model is essentially this, that we, on behalf of the bioinformatics community, can load this information into Wikidata. The rationale for Wikidata, it's run by the same group that runs Wikipedia, so this actually becomes an important resource for all of the 287 language-specific Wikipedias. So this is one resource that consumes Wikidata. But importantly, right, there's actually the ability now, as we in the bioinformatics community can also query and consume Wikidata content. The bioinformatics community isn't the only community here. There are many related communities that are also adding relevant information. So there's people who are adding toxicology information, people who are adding epidemiology information. Again, the point is that Wikidata is a platform that anybody can edit. Anybody can edit from importing very, very large data sets all the way down to single facts that are missing within Wikidata. And the point is that by putting it all in a centralized database, we all have access to be able to query it. Just to give you a sense for some of the type of integrative queries that I think we are enabling, right? So let's come back to this prototypical example that I started with. Show all tyrosine kinase inhibitors that are used to treat hematological cancers. I don't expect you to read this. I can barely read it. But this is the query, essentially, to query Wikidata for that answer. Okay, so this is a query language called Sparkle. It's a standard of the semantic web. But point being that once you can actually make the query, right? Once you get beyond the needs to go in the literature and spend hours and weeks on end pouring through review articles, then we can start to build analyses on top of queries like this. Okay, so show all tyrosine kinase inhibitors that are used to treat hematological cancers. Show all human membrane proteins associated with colorectal cancer. Again, a slightly longer, but still computationally tractable query. Show all monoclonal antibodies used to treat melanoma. Pretty simple query. We've been prototyping more and more use cases in the microbiome space. So, for example, show all organisms known to reside in the urogenital tract and can biosynthesize indole. This was actually a use case done by one of my postdocs, Tim Putman, who's around here somewhere, I'm sure, who worked in the chlamydia lab and looking at secondary infections that could co-occur with chlamydia. So these are all queries that I think are not easily possible using the existing tools, and I think if we can continue to grow the sum total of knowledge in Wikidata, we'll be in very good shape moving forward. Okay, so Wikidata enables this massive graph. Just like Wikipedia, Wikidata is essentially scopeless. So you can drift from Relin into, again, epidemiology, toxicology, all the way out to, in this massive neck, to Barack Obama and everything else in Wikipedia. There's a computational platform here where you can actually query. And just to say that even though Wikidata is 2, 3, maybe 4 years old, already there are encapsulated about 25 million items in Wikidata. 100 million statements are there for a total of about 1 billion triples. So 1 billion subject, object, predicate, right? So huge amount of data in there already, and it's continuing to grow as we get sort of the crowd effects operating here. Okay, that's part one. I hope I've convinced you that there is value in having centralized resources for biomedical linked open data. And this is really typifies our crowdsourcing activities among essentially domain experts, right? Scientists and other professionals. What I want to turn to for the second half of this talk is essentially this field of citizen science. And again, I defined citizen science before, but I'll just define it once more. Citizen science is the idea of building a platform, researchers building a platform that allows anybody in the community to contribute without any specialized training or education. Essentially, we'll build their training up from ground zero. There's a long history of citizen science efforts. I'll allude to some of the other projects a little bit later. But again, our idea is, can we apply citizen science to this challenge of knowledge management? Okay? So the first question along those lines is this. Can a group of non-scientists collectively perform concept recognition in biomedical texts? Okay, so remember, concept recognition was the first step in our two-step process, right? Identify the concepts, identify the relationships between the concepts. So far, we've mostly focused on the first one, okay? Can we do this among non-scientists? To answer this question, right, we used a platform called Amazon Mechanical Turk. The only thing you need to know about Amazon Mechanical Turk is that it's a platform that allows you to engage large communities of individuals for essentially micropayments. You pay them a little money, they do a little work for you, they're generalists. And so it's a good way to get an approximation of this non-scientist citizen science community. Okay? So we need to compare citizen science work and crowdsourcing work to the experts. And for that, we need a gold standard. Okay, so our gold standard was this data set of 593 PubMed abstracts, from which experts had identified almost 7,000 mentions of disease concepts. Okay, so this is the gold standard against which we were going to compare our crowd workers. If you send a professional biocurator, generally a PhD level scientist, against this problem, you see an average F-score of 0.87. Okay, so an F-score is merely an accuracy score. It balances precision and recall. It's a range of 0 to 1. Higher is better. Okay, that's all we need to know about the F-score. If now you pay a crowd, a member of the crowd, right, from the Amazon Mechanical Turk, no special prior training, and you set them along the same task, you see an F-score of 0.78, so a significantly worse F-score. Okay, that's not surprising. This person has a lot of biomedical training. But what we find is that if you aggregate the efforts among many independent votes, right? We're not showing this to one member of the crowd. We're giving it to many people in the crowd, and essentially you average their results. What you find is you get essentially equivalent results. Okay, for this particular task, the flipping point was about 6. So 6 people from the crowd was approximately equal to 1 PhD expert-trained biocurator. So although I don't like to emphasize the cost equation very much, I will just point out that using the Amazon Mechanical Turk platform, this process of going from these, of biocurating these 593 abstracts took approximately 9 days. Then we were able to engage over 140 people, and it cost a few $100. In contrast to the team of expert biocurators that did it part-time over an entire summer. Okay, so now the question is, okay, we've shown that we can pay people to do this biocuration task at a pretty good accuracy. But the question is, does it scale, right? Does this platform scale to biocuration that we would want to do writ large? And just to, do the back of the envelope math, suppose we want 1 million articles, again, that are published each year. Suppose instead of 6 people, we want 10 people to be extra sure. We don't want to do just disease recognition. We want to do other types of tasks as well. And this is what we paid for each unit of work. You get down to about $2.6 million per year, which actually I think is pretty reasonable. But we've yet to convince NIH to give us this grant. Until then, right? We turn back to the idea of citizen science, right? We've shown using the experiment I just described that people are capable of doing this biocuration task, and now the question is, can we actually get them to volunteer their time to do it? Okay, to do it essentially for free. So we created this application called Mark to Cure. It's at marktocure.org. This is what it looked like when we launched this at the beginning of last year. And our goal was essentially to replicate exactly the experiment that we did in Amazon Mechanical Turk, where the only difference is instead of paying money, our plea is actually simply the request, help science, please. So this is how things shook out. It took a little longer because we were building our community from the ground up. We had approximately the same number of workers. The cost involved, the nominal cost is zero. Obviously, we spent quite a bit of resource to build the platform, but once you've done that, then the cost is zero. And we get roughly equivalent accuracies. And I think we can actually push this up through what we learned in terms of how we do user training and user education. Okay? So what I think this experiment shows is that not only people, crowdsourcing, members of the crowd are able to do it, they're actually willing to do it, willing to do biocuration tasks for free under the auspices of helping biomedical research. Now the question is, does citizen science scale? Okay, so again, I won't walk through this in detail, but just to say, if you did the number of annotation events you'd want per year, divide that by the rate that we saw community activity in our beta experiment, you are able to calculate, ballpark, the number of volunteers you would need to actually scale up to the 1 million articles per year. And the number you come up with is roughly 16,000 volunteers. Sounds like a lot of people. We have roughly 1,200 so far. But just to point out that in the ecosystem of citizen science efforts, right, 16,000 is not a horribly ambitious target. Zooniverse is sort of the granddaddy in this field. It was citizen science devoted towards annotation of astronomical images, right? Classifying galaxies by the shape and morphology and things like that. And they have over 1 million volunteers in their roles. Foldit is a crowdsourcing game focused on the challenges of protein folding. Incredibly successful. They have over 460,000 volunteers. iWire is an application to map the three-dimensional structure of neurons. 200,000 volunteers. Eterna is an RNA folding game. 37,000 volunteers, although I'm sure that's gone up since I last updated it. So this idea of getting to 16,000 people is not terribly daunting, right? The idea is if you build a platform that is engaging to scientists, there are a large number of people who will participate and contribute their time. Okay, so now we have this hammer. Where are we going to apply it, right? We come back to the second case story I told you about, right? Let's think about nGLI1, right? Suppose we have nGLI1 at the center of this network. We want to mine the literature around nGLI1 and find the related concepts, right? The genes, the drugs, diseases, the variants. And from there, we can continually building out more and more. And the idea is that by building out the map, potentially we can identify the avenues of research that could lead to the types of transformative areas of research, or in a best case scenario, right, drug repurposing candidates that could work for NGLI1 deficiency. So we rebranded our Mark to Cure site, focused around this challenge of NGLI1. We have an amazing partnership with the NGLI1 community and Matt Might and his family. And again, this is the use case, or this is our first entry point into real discovery-oriented work in rare disease space. So this is an ongoing experiment. This is a preliminary view of the NGLI1 focused biomedical network that is emerging. We've had, again, 1,200 contributors. We've processed 3,200 documents so far. Almost a million annotations. So this is all freely contributed work. I think this is, an interesting visualization. Let's see, in blue are the diseases, green are related genes and proteins, and pink are treatments or drugs. So one of the most interesting anecdotes actually comes out of that node right there. And that node there, for those who can't see it, is ACTH. So this is adrenocorticotropic hormone. Okay, so this is interesting because we showed this to Matt Might and we said, and that was the first thing that jumped out at him, because what he told us that, when he was younger, Bertrand was actually on ACTH as a therapy, and it actually worked great in terms of alleviating his epilepsy symptoms and many of his other symptoms. Unfortunately, you can't be on long-term ACTH therapy. The toxicities are too high. It's an immunosuppressive, and so they had to remove that therapy. But in a retrospective sense, I think this gave us some amount of a hint that we were hunting in the right space. And so really our focus now is moving from this retrospective validation into prospective suggestion of interesting avenues for research. We asked our Mark to Cure volunteers why they contribute, and I think this is a really great slide. It reflects a large number of different motivations, from people wanting intellectual stimulation, wanting to learn, to people who've had personal connections to disease, and especially rare disease, even if it wasn't NGLI1 deficiency that they had a connection to. Right? It's a powerful motivator. So, again, I hope I've convinced you that biomedical research really relies on effective knowledge management. The idea that, again, we need to take better care and better account for all the things that we know up to any given point of time. It references this idea of standing on the shoulders of giants. Right? The giants that preceded us have all put their the knowledge that they've uncovered in the biomedical literature, and we need to, I think, better leverage that. The last thing is just to thank all the people that are involved. I have a great network of collaborators around the world. Again, I've mentioned Matt and Christina Might and the NGLI1 Foundation on our Mark to Cure project, Paul Pavlidis and Lynn Schrimmel with our Gene Wiki and Wikidata work. We get and of course, all of the crowd volunteers, without whom none of this crowdsourcing ideas would actually work. Most of our direct funding comes from NIGMS, and now more recently through NCATS. We're lucky to be involved with a couple really interesting common fund projects from this Big Data to Knowledge, as well as a program on extracellular RNAs. Here are some of the collaborative grants that we've written and received with other folks here at Scripps. And lastly, it's the fantastic crew of group members that I've had a pleasure to work with. These are the giants on whose shoulders I stand, and very appreciative to all the people who've been who participated. All the folks in green are current group members, and I've highlighted some of the Gene Wiki participants, as well as the Mark to Care participants or scientists with the stars. I've mentioned Chun-Li and Ben before, and together with Shirley, these three have been part of the lab since I arrived 5 years ago. So with that, thank you very much for your attention. Happy to take questions.